lecture, and in today's video lecture I will not be solving previous year's NEET questions. As you can see on your screen, we will be discussing countercurrent multiplication and exchange. Okay, and this is related to the topic excretory products and elimination. Okay, because I believe that many of you tend to get confused, like what does countercurrent mean? Okay, why is it there? Okay, so today I'll be clearing your doubts and I will explain countercurrent multiplication and exchange in detail. Okay, so let's begin. Okay, so in today's lecture I will be discussing countercurrent multiplication. We change this. Okay, so the first topic is countercurrent multiplication. Okay, then you have countercurrent exchange, and at the end I will be discussing urea recycling, sorry, which is related to countercurrent multiplication. Okay, and we'll see how. All right, so now before I begin explaining countercurrent multiplication, exchange, urea recycling, we need to understand why does this whole mechanism exist, okay, in the renal medulla. All right, so if you watched my previous video lectures, you would by now understand the meaning of concentrated urine. Okay, the formation of concentrated urine. Now it depends on our water intake. So the kidneys are designed in such a way that depending on our water intake and water availability in our body, it can excrete a concentrated urine or a dilute urine. Okay, can excrete a concentrated urine or a dilute urine. So if there is a lot of water, what happens is it tends to excrete that water out because we don't need it. Okay, but when there's a water deficiency, when there's a water deficit in our body, okay, especially during summers, okay, when we tend to sweat a lot, right, so we're losing out water from our body. So the nephrons are designed in such a way, the kidneys are designed in such a way that they will try to conserve the water. They will not allow the water to be excreted out. Okay, along with the solutes are being excreted out like they are, but the water concentration, the water availability will be low in the urine. Okay, it will try to reabsorb all the water out into the system, okay, into our body. So now how does a concentrated urine form? Okay, so the formation of concentrated urine, it includes hyperosmolarity in the renal interstitial medulla. Okay, this is one way in which a concentrated urine is formed. Okay, there's a hyperosmol region, okay, hyperosmol region in the renal interstitial medulla. Okay, so if you remember the structure of the nephron, it is divided between cortex and the medulla. You have the loop of Henle, right? You have the loop of Henle which goes down into the medulla region and the loop of Henle is responsible for creating that hyperosmotic region. Okay, this is the medulla. Okay, this is the loop of Henle, LOH. Okay, this region over here, this is the interstitial part. Okay, so a hyperosmotic, this part over here, this is the interstitial region of the renal medulla. Okay, and this is an hyperosmotic environment. Okay, it's an hyperosmotic environment, okay, which allows us to conserve water, okay, and in turn form a concentrated urine. All right, now this is one part, okay, how concentrated urine is formed. The other part is high ADH levels, antidiuretic hormone, okay, which is secreted from the posterior pituitary gland from the brain. So high ADH levels, when the ADH levels are high, okay, they act on the later distal tubule part. Okay, now this is the early distal tubule part and then the later distal tubule part, and the cortical collection tubule, the medulla collection tubule. Okay, and then it forms the collecting duct. Okay, so in the later distal tubule part and in the cortical collection tubule and in the medulla collection tubule, there are receptors for ADH. Okay, so most of the water is actually being reabsorbed in the later distal tubule and the cortical collection tubule. There is some water reabsorption happening in the medulla, okay, but most of it is happening in the cortical collection tubule and the late distal tubule. Okay, so there water does not just pass easily, they don't just diffuse via osmosis just like that. Okay, they are dependent on the level of ADH in our body. Okay, so for example, if we have to excrete a concentrated urine, okay, now in our system, the system always tries to maintain a homeostasis, always there's a balance created. Okay, so now if the system wants to excrete a dilute urine, okay, what will it do? It will reduce the levels of ADH. We don't want ADH because ADH tends to reabsorb the water. Okay, it creates water channels by which water gets reabsorbed. Okay, but we don't want that, so the ADH levels reduce. Okay, but if you want a concentrated urine like we are talking about today, if you want a concentrated urine, what will happen? Then the ADH levels increase and the ADH begins to reabsorb water. Okay, water over here, water over here. All right, it begins to reabsorb water. Okay, so another way that concentrated urine is formed is via high ADH levels. Okay, now lastly, urea recycling. Now urea recycling is very much related to the hyperosmol region of the renal interstitial medulla, and like I told you, loop of Henle plays an important role. Okay, we'll come to that. That is what countercurrent multiplication is. Okay, so loop of Henle plays a very important role in creating this hyperosmotic environment. Okay, it creates this hyperosmotic environment. Okay, urea recycling, when urea, by the time it reaches this medulla collection tubule, is very concentrated. Okay, now usually in our body around 20 to 50% of the filtered urea load is excreted out. This is a range. Okay, but some of it gets reabsorbed back into the body. Okay, some of it gets reabsorbed back into the body, okay, via diffusion. So the urea concentration over here is very high and tends to get reabsorbed back into the system. So this urea keeps recycling over and over again while there is more urea coming in. Okay, there's more urea coming in from here and it keeps recycling. Okay, we will come to that, why this urea recycling needs to be there. Okay, so these are the three reasons or the ways, sorry, by which concentrated urine is formed. Okay, so let's move on to our next step. All right, so you have countercurrent multiplication. Okay, so before you see a diagram of the loop of Henle. Now previously in the previous slide I told you that a hyperosmotic environment is maintained, right? And hyperosmotic environment is maintained over here, okay, by the loop of Henle. Okay, and this helps in the conservation of water. Okay, this helps in the conservation of water. So now this hyperosmotic environment is regulated, or it is formed by the loop of Henle. Okay, and we'll see how. So now the countercurrent multiplication is a system by which the hyperosmotic environment is maintained. Okay, it is created rather. Okay, hyperosmotic environment is created by the countercurrent multiplication system. Sorry, environment is formed or created. Okay, yeah, so the hyperosmotic environment is formed or created by the countercurrent multiplication. Okay, now countercurrent multiplication is is just basically three simple steps. Okay, it is just basically three simple steps, and you don't need to complicate it. Don't overthink it, because if you overthink it, you will get even more confused. Okay, so it is just divided into three simple steps. We'll go to the first part. So now let's assume the loop of Henle, the osmolarity in the loop of Henle is 300 milliosmoles per liter, okay, which is the same as the PCT. Okay, let's begin. This is like the starting point. Okay, let's assume that the osmolarity is balanced in the interstitial region as well as the loop of Henle region. Okay, it is maintained at 300 milliosmoles per liter. Okay, now please go and read the functions of loop of Henle, PCT, DCT, because we need to understand this. Okay, where what is being reabsorbed, what is going in. Okay, now we have the ascending limb of loop of Henle. Okay, the ascending limb of loop of Henle, and that is not permeable to water, but it is permeable to solutes. That's sodium potassium chloride. So there's an active pump over here, sodium potassium chloride pump. Basically, sodium and chloride are responsible for creating the hyperosmolarity region in that interstitial medulla. Okay, so they pump out salts. Okay, they pump out sodium and chloride. They pump out sodium and chloride in the ascending limb of loop of Henle. Okay, so automatically the osmolarity will change. Okay, automatically osmolarity will change. That means now from 300 it has gone down to 200. Why? Because salts are moving out. Now wherever there is more salt, there is more osmolarity, right? Hyperosmolarity region means what? Means there's lots of salts in it. Okay, there's urea, there is sodium, there's chloride, there's a lot of salt. There's a hyperosmol region. Okay, so these salts move out into that interstitial medulla region. Okay, now once these salts move out, this osmolarity will decrease, whereas this osmolarity will increase to 400. Now 300 has become 400 and 300 has dropped down to 200 over here. Makes sense, right? Now, but there is an imbalance over here. Okay, always the body tries to balance. Even osmolarity, it will try to balance. If there is salts coming out, there will be water also coming out to try to balance that situation. Next step. Now salts have come out. Now we know that the descending limb of loop of Henle is permeable to water, right? Descending limb of loop of Henle is permeable to water, but not permeable to salts. So water will tend to move out. Water will tend to move out over here. So what happens over here? So 300 will become 400, right? Because now there is more salt content inside the descending limb of loop of Henle, right? So water is coming out, that means salt is still remaining inside. That's why you see this dark liquid over here, this dark yellow color. That means it's more concentrated, right? It's more concentrated. Over here it is light because salts have moved out. That means there's liquid over here, this water over here. So that means it's less concentrated. Okay, so now water has moved out into this region. Okay, so now it has balanced itself out. Now 400, 400, 400, 400, 400, 400. Okay, fine. So solutes moving out from the ascending limb of loop of Henle and water moving out from the descending limb of loop of Henle. So you have one side you have salts coming out, one side you have water coming out. Okay, that's how the hyperosmol region is being created now. Now, obviously now salt has moved out, water has moved out, but PCT is still dropping in the filtrate. PCT is still working, right? PCT, proximal convoluted tubule, that's what I mean by PCT over here. Sorry, PCT is still dropping in that filtrate into the loop of Henle, right? So there's more filtrate coming in filled with solutes. There's more filtrate coming in filled with solutes. So what happens is this region over here is getting pushed down. This region, this concentrated region, now water has moved out, so this region has become concentrated with solutes. So this region gets pushed down, okay, to make space for the filtrate that's coming in from the proximal tubule. Okay, to make space for the filtrate that's coming from the proximal tubule, this liquid or this concentrated liquid gets pushed down. Okay, that's why you see this 400 has now taken up space over here in this U-shaped region. Okay, the U-shaped pin that is there. Correct? So now this is the fourth part. Fifth part, again it is a repeat of the same steps. Again now what solutes are moving out from this is the ascending limb of loop of Henle, permeable to solutes. So solute is moving out, solute is moving out. Again this osmolarity has decreased from 200 to 150. From 400 it has gone down to 300. So osmolarity, because salts are moving out. Wherever there's high region of salt, the osmolarity will be high in that region. Okay. So now the job is to create a hyperosmol region in that interstitial medulla, right? So salts are moving out. So now here, so 200. So yeah, so 300 has now this has become 500 over here. Clearly, right? So salts are moving out over here and this has become also 500, right? Now similarly the same step, water will move out to balance that. Water has to move out. So water will move out to balance that. So the insides become 350, 500, 500, from 400 to 500, 400 to 500. Okay, 300 becomes 350. So see, this has become even more yellowish. Okay, so it has become even more concentrated. Water has moved out. So this region has become even more concentrated. Correct? Again this step 4 to 6 is repeated multiple times. Same thing. Okay, again proximal tubule is pushing down the liquid in. Okay, so again this region will go down. This region will get pushed down, so everything will come 500, 500, 500 over here, right? Again the same thing, solutes will move out, water will move out, again proximal tubule will push in the liquid. Now this step is being repeated multiple times. That is all. Okay, that is all that is happening till a region, a situation occurs where this concentration increases up to a maximum of 1200 milliosmoles per liter. The maximum that it can concentrate its region is 1200 milliosmoles per liter. That's where it stops. Okay, that's where it stops. Okay, so this repetition of the same steps, the proximal tubule pushing in the fluid and loop of the ascending limb of loop of Henle is pushing out the solute, the descending loop of Henle is pushing out the water to balance that osmolarity. The repetition of the same steps is countercurrent multiplication. Okay, you see the osmolarity is being multiplied with every step. With every step the osmolarity is being increasing. Okay, countercurrent, why? Because they are moving in opposite directions. Okay, they're moving in opposite directions. Okay, they are countercurrent with each other. The descending limb, ascending limb, descending limb, ascending. So this U-shaped structure that is there is very helpful in creating that hyperosmol region. That is why, or that is how rather, hyperosmotic region is created in the kidney. That is all. There's nothing very complicated. The first step, let's say we'll start from the ascending limb of loop of Henle. Ascending limb of LOH, right? The solute transport to the interstitial medulla. That's the first step. Second step, descending limb. Okay, descending limb is permeable to water, so to balance that osmolarity, again it will send out water from the descending limb of loop of Henle, right? So it will balance the, so that means this 300 will become 400, 400, 400, it will balance, right? So this region is becoming more concentrated. Again then PCT will drop in. Okay, PCT, so let's say water transported or diffused, rather, diffused to the, this is also not diffused, to the interstitial medulla, diffused to the interstitial medulla. Okay. So again, then PCT is pushing in that fluid back in. So third step is PCT. PCT's flow of filtrate into loop of Henle. Okay, so PCT's when PCT drops in its filtrate into the descending loop of Henle, so whatever liquid is there in the loop of Henle will get pushed down into that U-shaped region. Okay, it stays there like that. Okay, it stays there like that. And again the same protocol happens, the same method happens in the ascending limb of loop of Henle transports or diffuses its solutes out, and then to balance that osmosis, the water also gets diffused out from the descending limb of loop of Henle. Again then PCT pushes its fluid in. That is how a hyperosmotic region is created. Okay, this is called countercurrent multiplication. You can see the osmolarity is being multiplied. It's increasing. Okay, it's increasing with every step. Okay, it is increasing with every step. Okay, if you repeat the same steps over and over and over and over and over again, the hyperosmolarity will increase till it reaches a state of 1200 milliosmoles per liter. It has to balance, right? So it reaches a state of 1200 milliosmoles per liter. Okay, so basically this is what countercurrent multiplication is. Okay, it is a result of 3 steps that are happening. Okay, clear? So countercurrent multiplication, through this we have created a hyperosmotic region in that renal interstitial medulla. Okay, now let's go to the next step. Okay, so the next step is countercurrent exchange. Now we discussed countercurrent multiplication, right? The hyperosmotic region is created in the interstitial medulla, but now the sodium chloride is present there, urea is also not present there, all these solutes are present in that hyperosmotic region, but there should be a way to maintain that, right? There should be a way to maintain that, because if solutes are being pushed into the diffused into the hyperosmotic, the renal interstitial medulla, they don't just stay there for long. Okay, if they stay there for long, they begin to dissipate. Okay, they tend to disappear, because we have to maintain a hyperosmotic region. So a countercurrent exchange system is there. A countercurrent exchange system is present there, which helps to maintain that hyperosmotic region. Okay, so this is called countercurrent exchange, and it is possible with the help of vasa recta. Okay, it is possible with the help of vasa recta. Now how does that happen? Okay, so you have the loop of Henle. Okay, and then you have the vasa recta. This is also a U-shaped structure. Okay, anatomically this U-shaped structure is very important in maintaining the hyperosmotic region. So now when obviously the cells in the kidney need to receive blood, right, for its metabolism, correct? So there is a flow of blood into the kidney cells. Okay, but this flow of blood is very slow, it's very sluggish. Okay, this flow of blood is very sluggish. So there's very slow diffusion. Okay, there's very slow diffusion of solutes and water. There's very slow diffusion of solutes and water. It's not happening very quickly. Okay, so this in a way helps to maintain that hyperosmotic region. It's not quickly that solutes are moving and solutes are moving out, water is moving and water is moving out. No, it has to maintain a particular osmolarity also, right? It has to maintain that 1200 milliosmoles per liter. So this flow over here is a very slow flow. Okay, very sluggish flow. Now we know solutes come out from here, right? Solutes come out from here, then solutes have to go. This is the ascending limb of loop. So filtrate is moving like this. So filtrate is moving like this. So solutes are moving out from here. Solutes are going into this region also. but this is a very slow region. Okay, this is a very slow region. So solutes moving out from here will go into this region. Water moving out from here will go into this region. Okay, this is again a counter current exchange happening, but there are two different directions. Again, this is also moving in two different directions. This is also moving in two different directions. So solutes that are coming out will slowly then move into the vasa recta. Okay, water that is coming out will slowly move into the vasa recta. Okay, so that way countercurrent exchange does not help in creating a hyperosmolarity region. It has no contribution in that. What it does is just maintains it. That is all. The exchange system is just to maintain that. Okay, that exchange, that hyperosmolarity region. Okay, hyperosmolarity, what we saw in countercurrent multiplication, yes, that is a very important role. The ascending limb of loop of Henle and the PCT that's dropping in, that whole cycle is very important. The multiplication is very important in creating the hyperosmolarity region. Okay, whereas exchange just helps in maintaining that system. Okay, it needs to maintain that system, because if solutes and water and all tend to remain there for a long period of time like that without being pulled in, and so that there is chance for more solutes to come out, okay, then those solutes then begin to dissipate. When we don't want that to happen, then the whole system will break down. Okay, so now to maintain that, it needs to go out also, so that there's space for more solutes to come in. There should be space for more water to come in also, right? Because if only solutes remain there and are not pushed up, the water keeps pushing in or diffusing into that system, then the whole system will break down, it will become dilute. We don't want that. We want to maintain a concentrated region, and that is obviously maintained at 1200 milli. The maximum is maintained at 1200 milliosmoles per liter, and that is at the tip. That is at the tip over here. Okay, that is at the tip over here. All right. So like you can see over here, as solutes are moving in, okay, solutes are moving in, obviously the osmolarity, and water is moving out, so obviously the osmolarity is being maintained here also in the vasa recta. This is a very intelligent system. Okay, that not only in the loop of Henle that osmolarity has to be maintained, but also in the vasa recta. Okay, because a balance always has to be created. All right, so as the blood is moving down, solutes are moving in, so that is contributing to the osmolarity inside. Okay, but water also tends to move out. Okay, and the water is then being reabsorbed over here. Okay, and the solute moves out again. So what it does is it creates a similar osmolarity region in the vasa recta also, just like in the loop of Henle, just like in the interstitial medulla. All right, so everything is balanced out. Okay, the vasa recta is also balanced out. This is the same osmolarity. Okay, as it's moving down, solutes are moving in, the water also is moving out. The water doesn't stay here for long, water is then getting pulled in over here, right, and the solutes are getting pulled out over here. Okay, so this maintains a hyperosmolarity region. That is all the job is of the vasa recta over there. Okay, the countercurrent exchange. This is called countercurrent exchange system. All right, so now what we have seen over here is how a hyperosmolarity region is created in our system. Okay, we have the countercurrent multiplication, we have the countercurrent exchange system, which I said is for maintaining the hyperosmolarity region. Okay, now obviously now the hyperosmol region is created along with ADH hormone that is there. So whenever you want to create a concentrated urine, there are two factors by which a concentrated urine is created. Hyperosmolarity, which helps in the diffusion of water. Okay, it helps in the diffusion of water from the filtrate into the interstitial medulla. Okay, and by then the water is being reabsorbed into the vasa recta also. Okay, hyperosmolarity is one way by which water diffuse. Also, it depends highly on ADH levels. it also depends highly on ADH levels. Okay, now in the later part of DCT and in the cortical collection tubule and in the medulla collection tubule, ADH attaches. Okay, there are receptors over there. ADH will attach whenever you want to create a concentrated urine. Okay, now ADH creates these water channels. Okay, ADH, if you don't follow what I'm saying, is please watch my previous lecture where I have explained the function of ADH. Okay, that's part 3 of excretory system. So ADH creates this environment, water channels called aquaporins. Okay, by which the water then diffuses out. Okay, water then diffuses out. Okay, into the interstitial medulla. Oh, these two are responsible for creating concentrated urine. Okay, so when ADH is present and a hyperosmolarity region is present, ADH attaches itself, creates the water channels by which water will then get reabsorbed back into the region, okay, into our system, into our interstitial fluid, okay, interstitial medulla region. Okay, this happens via the process of diffusion. So water channels are just a way of creating that area, those channels by which water can get reabsorbed back into our system. Okay, and the hyperosmolarity creates the region by which water can easily diffuse. Okay, water can easily diffuse into the region. Okay, so there are basically two factors by which concentrated urine is formed: hyperosmolarity of the region, which I've already explained, countercurrent multiplication and exchange, and ADH, which you need to study. It's an antidiuretic hormone which helps in creating that environment or the place or a region by which water can be reabsorbed into the system. Okay, so let's move on to the next. Okay, so now we come to the last part. Okay, it's called urea recycling. Now, initially, in the initial part of the lecture, I already explained to you that a hyperosmotic region is created, okay, via which, with the help of ADH, water get diffused into the interstitial medulla and then into the vasa recta and back into our system, okay, which is what we want. But urea also plays an important role, okay, it also plays an important role and it contributes to around 500 to 600 milliosmoles per liter of that concentration. Okay, so urea does play an important role along with sodium chloride and water and all that is happening in the countercurrent multiplication. But urea's recycling, what basically urea does is just adding to that hyperosmotic region, that is all. Okay, it is only adding to that hyperosmotic region. And that urea along, now see solutes, like I said, solutes in water, they don't stay there for long. There's a exchange system where vasa recta is then pulling out the solutes and pushing in the water and pulling out the solutes, and that exchange system is happening, right? And urea in the same way does not stay in the region over here. It'll get recycled over here. It'll get recycled in that system, in the nephron. Okay, it gets recycled in the nephron. So it is a way of maintaining hyperosmotic region. I hope you understand what this hyperosmotic region means, right, in terms of osmosis, right? So if you have a cell and there's a hyperosmotic region outside the cell, okay, region outside the cell, what will happen? Water will move out, okay, and the cell will shrink. Okay, if you have a hyperosmotic region outside the cell, water will move out and the cell will shrink. Okay, so basically it is the same idea over here. This is, but with the help of ADH. Okay, with the help of ADH. All right. Sorry. Okay, so you can see the nephron over here. So now nephron is moving down this PCT. Now some reabsorption of urea is happening here. Okay, some reabsorption of urea is happening here, but It is not enough. It is not, because urea is not very permeable over here as compared to water. Okay, water is more permeable in PCT as compared to urea. So there's lots of water moving out, but urea not as much as water. So it remains concentrated. Now you understand the meaning of concentration. Water is moving out, so urea becomes concentrated in that region. Okay, so this is what we want to achieve. We want to achieve a concentrated urine where water is moved out, but the solutes are excreted like they should be, right? So over here, urea is being reabsorbed, but water is more permeable as compared to urea. Okay, anyway, so urea moves down. Okay, urea moves down the descending limb of loop of Hindley. All right, so now we know the descending limb is permeable to water, so water is again moving out over here. So what happens, the urea concentration keeps increasing because water is just moving out. So urea, we are only focusing on urea, so urea concentration keeps increasing. Okay, furthermore, there are some transporters over here. Okay, there are some transporters over here in this U-shaped region over here in the loop of Henle, in the medulla, the inner medulla region. Okay, which further then increase the concentration of urea. Okay, they further increase the concentration of urea. There are some transporters, as you can see, it is what it is coming from this collection tubule, the medulla collection tubule. Okay, which is further increasing the urea concentration. Now urea moves up. You see this part, this part, this thick brown part over here, this is where urea will not get reabsorbed. Urea will remain as it is. Okay, urea will not get reabsorbed or nothing will happen to the urea. Okay, so urea will just remain as it is till it reaches down, okay, into the medulla collection tubule. Okay, but what happens in this region? Water is getting reabsorbed, right? So the ADH is moving in, ADH is moving in, forming these water channels in the late distal tubule, in the collection tubule, in the medulla tubule also. But there is less over here, but more in the collection, the cortical collection tubule and the late distal tubule. ADH is attaching itself, so water is moving out. Okay, via that ADH level of ADH and the hyperosmotic region that is there. So water is moving out. So then what happens? Water is moving out, urea concentration is increasing because of that. Okay, as water moves out, urea concentration is increasing. Okay, till it reaches 300. Okay, it is a lot. Okay, it is a lot. Okay, and then there are some transporters over here in this medulla region which allow the diffusion of urea, okay, from high concentration to low concentration of urea. So there are some transporters over here which allow the urea diffusion from high concentration to low concentration into this interstitial medulla. So what is happening? Already there is salt content, urea is being added in. What happens? The hyperosmolarity increases. So what it does is it is increasing the hyperosmolarity region. Not really maintaining it, I would say increasing. Sorry. Because maintaining it is done by the vasa recta. It is increasing that hyperosmolarity region, okay, in this region, in this space over here. Okay, there are some transporters here which allow the diffusion. Okay, there's already high concentration of urea, so high to low concentration diffusion will happen. Okay, so urea will move in, but there are some transporters over here also which accept this urea. Now again, this region is still higher compared to the urea over here, urea concentration in this region, correct? So urea will move in from high to low. Okay, diffusion is happening like this, and it again moves up. Again there is no urea reabsorption, nothing is happening over here. Okay, it is not permeable to urea at all, this region over here. Again it is going to the medulla region over here. Again the same thing happens, urea will move out from high concentration to low concentration. Not all the urea is not being reabsorbed back, we don't want that. There is some of it that is moving out, that is excreted out of the system. Yes, okay, but there is most of it that is being reabsorbed. Okay, what is that? That is just increasing the hyperosmolarity region in the interstitial medulla. Okay, so urea moves from high concentration, low concentration over here, and then again high concentration to low concentration over here, and again it is again recycle. Now just imagine what we studied in countercurrent multiplication, those steps. When it is repeated over and over and over again, what happens? The hyperosmolarity is increasing there, right? The osmolarity is increasing there. Similarly, we are achieving the same goal over here. We are recycling urea again and again and again and again and again in this region. Okay, we are recycling the urea again in this region, so its concentration keeps increasing. Okay, obviously urea is coming in from here also, right? From the PCT down to the descending limb of loop of Henle, already its concentration is increasing here. Add to that, this urea is also being diffused into this ascending limb of loop of Henle. Again, if you just keep repeating the same process over and over again, the concentration will increase. Okay, the concentration will increase, and this job is nothing but just to increase the hyperosmolarity region of that interstitial medulla. Along with the solutes, that countercurrent multiplication, that part is happening. Along with that, this urea recycling is also happening. That is all. Okay, that is all that's happening. So in conclusion, let's revise what we have studied. Now now not even let me not go into countercurrent multiplication as yet. Let me just talk about concentrated urine. Okay, I said two things, just two things. Remember that it's with the help of ADH and with the help of hyperosmolarity region in the medulla. Okay, urea recycling just adds onto that hyperosmolarity region that is created by countercurrent multiplication and exchange system. Okay, that just adds to that urea recycling. So at the end of the day, what happens? Water is being reabsorbed, okay, with the help of ADH. So ADH does play an important role. Okay, ADH does play an important role, and it creates that environment, the region, the water channels through which water will get diffused out. Okay, through which water will get diffused out, okay, into that hyperosmolarity region. Okay, this is what we want. Diffusion is happening, but that is only with the help of ADH, and that only happens in this region. Okay, now this region is not just permeable to water via osmosis like it happens in PCT or anywhere else, or in the loop of Henle or anywhere else. No, this region will only be active when there's ADH present. It just the water just doesn't leave like that just by osmosis. Please remember that. Okay, this could be a very good question in your exam also. Okay, it's only with the help of ADH which creates that the water channels via which the water will then get diffused out, okay, into our system, okay, which we want. Okay, and urea recycling comes later, but countercurrent multiplication exchange system. Countercurrent multiplication is the main reason or the main way, a method by which a hyperosmolarity region is formed, it is created. Okay, while solutes moving in, water moving in, PCT is pushing in the filtrate down, again same solutes, water, PCT is pushing down. These three steps, that is all. Okay, that is all that countercurrent multiplication is. This is a repetition of these three steps. All right, countercurrent exchange is just a way of maintaining that. We don't want the solutes to just dissipate, so there's a very sluggish flow of blood, okay, which just slowly then diffuses the solutes in, pushes the water out, diffuses the solutes in, diffuses the water out. Okay, and then again it takes the water in, pushes the solutes out. Okay, there's a constant exchange that is happening, because then the solutes and water and all tend to remain there. If solutes begin to dissipate and water just remains there, the whole system will break down, right? So we just want to maintain an exchange system by which our hyperosmolarity region is maintained. That is all. Urea recycling, simple. Urea recycling is just to add on to that hyperosmolarity region. Okay, urea recycling just adds into that and contributes to about 500 to 600 milliosmoles per liter. Okay, so it's a very big portion. All right, obviously not all the urea is being recycled out, right? So 20% to 50% does get excreted out from that filtered load. Okay, and the rest they get that gets recycled in that system, in the nephrons. Okay, in the nephrons. So at the end of the day, what you get is you get a concentrated urine. Okay, so our bodies are designed in such a way that we try to conserve water as much as possible. Okay, our bodies have to create a balance in the system. Okay, always a homeostasis region is always created in the system, by which we don't have too much salt and we don't have too much water, right? If you have too much water, what happens, it'll just push out all the water, right? It'll tend to push out all the water. But what if you've not drank water? What if there's deficiency in the water in the system? Because our cells need water, we need water to survive, right? Our metabolic systems also need to work. So for that, it will tend to conserve water as much as possible. It tend to conserve water, so at least our metabolic systems are functioning like usual. Okay, so basically this is what I wanted to explain today, and my focus was obviously on countercurrent multiplication, exchange, and urea recycling. Okay, and yeah, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new, because I understand that this topic is a little tricky, but it is at the end of the day, it is a very simple topic. Like I said, it is very simple. Don't overcomplicate it, just try to simplify things. Okay, and please like, share, and subscribe to MG NEET Biology if you've enjoyed today's lecture. And also, I'm available on Facebook, Twitter, and Instagram, so you can follow me there for updates. All right, and I'll see you in the next video lecture. Okay, bye.